Hello student. Today we will discuss about molecular basis of genetic codes. In this lecture, we will take a closer look at the genetic code which allows DNA and RNA nucleotide sequence to be translated into the amino acid they represent. I am Dr. Ashishan. I welcome all of you to another interesting and important session. So student, my question to you all. Have you ever written a secret message to one of your friends? Yeah, obviously. You may have used some kind of code to keep the message hidden. For example, you may have replaced one letter or word with number or symbols, following particular sets of rules in order of your friend on the other end to understand the message. They would need to know the code and apply the same set of rules in reverse to figure out what you had written. As it turned out, decoding message is also a key step in gene expression, the process in which information from gene is used to construct a protein. However, the instruction for building a protein encoded in DNA. By the end of this session, you will be able to answer how is the information in an messenger RNA sequence decoded to make a polypeptide, and how group of three nucleotides called codon specify particular amino acid. Gene is the basic unit of all molecular and genetic chemistry. So genes that provide instruction for protein are expressed in two steps. First is transcription and second one is translation. In the transcription, the DNA sequence of a gene is rewritten using RNA nucleotides. In eukaryotes, the RNA must go through additional process to become messenger RNA or mRNA. In the translation, the sequence of nucleotide in messenger RNA is translated into a sequence of amino acid in polypeptides that is called protein or protein subunits. Cells decode messenger RNA by reading their nucleotide in group of three called codon. This process called central dogma of molecular biology, described by two-step process, transcription and translation. The term codon is given by George Gamow. He is Soviet-American theoretical physicist. Genetic code is a dictionary that corresponds the sequence of nucleotides and sequence of amino acids. The letters A, G, T and C correspond to the nucleotides from in DNA. They are organized into codon. The collection of codon is called genetic code. For 20 amino acids, there should be 20 codons. Each codon should have 3 nucleotides to impart specificity to each of amino acid. For a specific codon, one nucleotide makes 4 combination, 2 nucleotide makes 16 combination and 3 nucleotides make 64 combination. 4 into 1, 4 combination, 4 into 4 16 combination, 4 into 4 into 4 equal to 64 combination. So there are possible combination of codon and their expression to the genetic information to synthesis a particular amino acid. So student, it is clear. Each codon consists of 3 bases. This is called triplet. So there are 64 available codons. But in out of 64, 61 codon code for 20 amino acids found in the protein, but 3 codons do not code for any amino acid. These 3 codon, they do not code any amino acid called nonsense codon, termination codon or stop codon. The stop codons are UAA, UAG and UGA. The ribosome falls and falls of the messenger RNA. The name of 3 nonsense stop or termination codons are UAA is Oka codon, UAA is Amber codon, and UGA is known as Opal codon. There are some important properties of genetic code. First, codes are triplet, commaless, non-overlapping, degenerate, universality of codon, and the codes are non-ambiguous, chain initiation and chain termination codon. So we will discuss about properties of code one by one. First property of genetic code is triplet. Total 64 codon are available, out of which 61 code for amino acids and 3 codon do not code for any amino acids. Hence, they function as stop codon. Example: UUU for phenylalanine and GGG for glycine. Second property is commaless. So code are commaless. The in between triplet codon, no letters are wasted. No punctuation are needed between any two codon. So the gene is transcribed and translated continuously from a fixed starting point to fixed stop point. Third property of genetic code is the code is non-overlapping. The all code are independent set of three ways. There is no overlapping. The code should be always read in group of three nucleotides from a fixed starting point. The three letters of codon are considered as unit and should be read as if they are exposed through a reading frame. The reading frame, the starting point is extremely important and this is called reading frame. Fourth property of genetic code is degeneracy. Amino acid can be coded for by more than one codon. This is called degeneracy of genetic code. For example, serine is coded by UCU, UCC, UCA, UGC, AGU and AGC. Fifth property of genetic code. This is unique property. That is universality of genetic code. The genetic code is universal. The same code apply in all kind of living organisms. Example: UUU code for phenylalanine in all living organisms. AUG is codon for methionine in mitochondria. The same codon AUG code for isoleucine in cytoplasm. With some exception, note the genetic code is universal. Sixth property of genetic code is non-ambiguous. Thus, one codon can not specify more than one amino acid. Mean one codon code for only one amino acid, hence it is specific and non-ambiguous. Seventh property of genetic code is chain initiation codon. The codon which initiate the process of polypeptide chain formation are referred as starting or initiation codon. For example, AUG is the starting codon and is present as triplet codon on all messenger RNA, and it code for amino acid methionine. Some exception with chain initiation codon. In few cases, GUG may be the initiator codon. The last property of genetic code is termination codon. The triplet code which is at the end of messenger RNA and does not specify for any amino acid and serves for termination of polypeptide synthesis is known as termination stop or nonsense codon. The example of termination codon are UAA, UAG and UGA. One more important characteristic or property of genetic code which is not listed above, this is polarity. The code has definite direction for reading of message which is referred as polarity. Reading of messages from left to right or right to left will be specified for different amino acids. For example, UUG stand for leucine and from right to left it is GUU, which stand for valine. One important hypothesis related with property of genetic code. This is vowel hypothesis. Crick postulates the vowel hypothesis in account for degeneracy of genetic code. According to vowel hypothesis, the first two bases of genetic code pair accordingly to normal base pairing rule, with the last two bases of anticodon base pairing at the third position of a codonist vowel. So this is the explanation of degeneracy of genetic codon. For example, although there are 61 codon for amino acid, what the number of transfer RNA is less is around 40, which is due to wobbling. Wobble means the unsteady movement from site to site. And what is anticodon? The base sequence of transfer RNA which pair with codon on messenger RNA during translation is called anticodon. There are some basic difference between codon and anticodon. First, codon could be present in DNA as well as RNA, but anticodon is always present in RNA, never present in DNA. Codon are written in 5 to 3 direction, whereas anticodon are usually written in 3 to 5 direction. Anticodon of some transfer RNA molecules have to pair with more than one codon. Anticodon discretely in the cell and not follow the sequentially arrangement. So codon are sequentially arranged in nucleic acid, whereas anticodon are discretely present in the cells. Codon define which anticodon should come next with an amino acid to create the protein strand. And anticodon help in bringing a particular amino acid at its proper position during translation. So what is the clinical significance of genetic code? So mutation can be easily explained by the using genetic code. So example of mutation, point mutation, it includes silent mutation, missense mutation and nonsense mutation. Second one is frame shift mutation. So first mutation is silent mutation. In this mutation, single nucleotide change in the same amino acid, example E to G. Same amino acid will be formed but defected amino acid. So this mutation always goes unnoticed. Another mutation is missense mutation. In this mutation, also single nucleotide chain, that is A to C, but synthesis of amino acid is totally different. So loss of functional capacity of protein. In nonsense mutation is responsible for stop codon generation. There is also single nucleotide change, that is C to A. So it synthesizes stop codon. In the messenger RNA represented by UAG, premature termination of chain may be incompatible with life. And in frame shift mutation, there is insertion or removal of base which can alter the reading frame, with resultant incorporation of different amino acid. And thank you. This is all over about genetic code.